Welcome back to this next video in which we are discussing influenza. So we're currently in the process of discussing the pathogenesis of influenza, and we're discussing the inflammatory response that is going to occur at the respiratory epithelia that are infected by influenza viruses. So let me remind you of where we've got to at this point. So remember, cells of the respiratory epithelium are going to die because of the influenza infection. When they get infected by the influenza viruses, they produce thousands of genetically identical viruses, but then they die because this exploitation kills them. When they die, they will release all sorts of molecules which we can collectively call DAMPs, which are telltale signs that something is horrendously wrong here. Those will bind to their respective pattern recognition receptors on the surface of sentinel cells, the two major examples of which are dendritic cells and resident macrophages in the underlying connective tissue underneath the respiratory epithelium, and these sentinel cells will then start calling for help. We need help to deal with whatever it is that has caused the death of these cells, and all of the help, all of the troops for fighting infection are in the blood. So, they release these help signals, these pro-inflammatory mediators, and I've given you 5 important ones here. The 2 most important ones to know the names of, forever shall their names be revered, are interleukin-1 and tumor necrosis factor alpha, but 3 other important ones are histamine, interleukin-6, and prostaglandin E2, and these are going to cause the inflammatory response, particularly the first 3, interleukin-1, tumor necrosis factor alpha, and histamine. They're going to cause the inflammatory response. We'll come on to what interleukin-6 and prostaglandin E2 do later on. Now, the inflammatory response is fundamentally a response of the microvasculature to these molecules, these 3 molecules, interleukin-1, tumor necrosis factor alpha, and histamine. Therefore, at the end of the previous video, we revised microvasculature blood vessels that are present in all tissues and will be present in this underlying connective tissue here, nearby the sentinel cells. So remember, we have the terminal arterioles here, which have this lining of vascular smooth muscle cells. Then we have our little capillary here, and then those capillaries reconverge up into a post-capillary venule, which is shown here. So now it's actually time to talk about the inflammatory response. So as I say, the ones that are going to cause this, of these mediators that I have listed here, are the first 3, interleukin-1, TNF-alpha, and histamine. They are major mediators that cause inflammation. They're going to cause the inflammatory response. So they're going to go over to these blood vessels. They're going to diffuse over in the extracellular fluid to these microvasculature blood vessels that will be in this underlying connective tissue here, and they are going to cause changes in those blood vessels that constitute the inflammatory response. So what are these changes? Well, remember what we want the blood vessels to do. We want them to start bringing troops from the blood into the extracellular fluid in this underlying connective tissue and the extracellular fluid of the epithelium here. Now, what are the troops that are in the blood? Well, there are 2 major categories of troops in the blood. There are the white blood cells, also called the leukocytes, and we're going to talk about 3 major types of white blood cell that in the inflammatory response, the acute inflammatory response, will be coming out of the blood and entering this infected tissue. And the other category of troops are proteins that are in the blood that are antimicrobial proteins. Major examples, of course, being the complement proteins. Now, these proteins are very effective against certain types of pathogen. They're not going to be particularly effective against influenza virus, but against other types of pathogen, they are very effective. And remember, this response is non-specific. It's not tailored to the influenza virus at all. It's the standard response that we initiate to all pathogens. So some of the troops that we bring into this area that is infected with influenza viruses are not necessarily going to be brilliant at dealing with the influenza virus, but they might be very good at dealing with some bacteria if they occurred at a site where there was a bacterial infection instead of the viral infection. Okay, so remember, this is a non-specific response. So what's going to happen is the interleukin-1, TNF-alpha, and histamine are going to work on these 3 types of blood vessel, and they're going to cause changes in the blood vessel. So let's start off with what they're going to do to the terminal arterioles. So they act specifically on the endothelial cells of the blood vessel. So they work on receptors that are on the endothelial cells of all three types of blood vessel, the terminal arterioles, the capillaries, and the postcapillary venules. And then by acting on the endothelial cells, they're going to trigger changes in the blood vessels. So let's start with the change that's going to occur in the terminal arterioles. The change that's going to occur in the terminal arterioles is that you're going to get vasodilatation. So I'll just jot this down here. So the components of the inflammatory response then. In the terminal arterioles, you're going to get vasodilatation, also often just called vasodilation. So I'll just call it vasodilation instead of vasodilatation. It's strictly speaking called vasodilatation, but vasodilation is gradually replacing vasodilatation because it's a more sensible word. And so it's going to cause vasodilation of these terminal arterioles, and this means that these terminal arterioles are going to dilate. They're going to get bigger. The diameter is going to get bigger. And the way that that occurs, of course, is that the vascular smooth muscle cells surrounding the outside of the terminal arteriole are going to relax so that the circumference of these rings of vascular smooth muscle cells gets bigger, and therefore the diameter of those rings of vascular smooth muscle cells is going to get bigger. So we need to somehow get relaxation of these vascular smooth muscle cells. And how is that going to occur? Because remember, I've said these molecules, interleukin-1, TNF-alpha, and histamine, act on the endothelial cells, not the smooth muscle cells. So what actually happens is these molecules are going to act on these endothelial cells, and I might just draw a little arrow here. So these are going to act on the endothelial cells of the terminal arteriole. They will diffuse over to the terminal arteriole. They'll make their way through the outer layers of the wall and into where the endothelial cells are, and they will act on receptors on the endothelial cell membranes. And what they will trigger is they'll trigger the endothelial cells to start producing another molecule in turn, which will be released and will diffuse back to the vascular smooth muscle cells. And this molecule is another prostaglandin by the name of prostaglandin I2, also called prostacyclin. So this has another name. Not only is it called prostaglandin I2, but it's also called prostacyclin. Okay, so just to summarize this again, say it again to make sure it's clear. Interleukin-1, TNF-alpha, and histamine, they act on the endothelial cells of the terminal arteriole and make them secrete another molecule called prostaglandin I2, also called prostacyclin, which they will release into the extracellular fluid and which will make its way back to the vascular smooth muscle cells, will work on receptors on the vascular smooth muscle cells and cause them to relax, and therefore the circumference of these rings of vascular smooth muscle cells will increase, and therefore the diameter of those terminal arterioles will increase. So this is overall then going to lead to vasodilatation or vasodilation. So this is the way in which interleukin-1, TNF-alpha, and histamine affect the terminal arterioles. They cause vasodilation of the terminal arterioles. Now, what on earth is the point of vasodilating the terminal arterioles in this area where the inflammatory response is occurring? Well, if you vasodilatate the terminal arterioles, that means that blood flow is going to increase to the affected area. More blood can come through the terminal arterioles, therefore more blood will go through the capillaries, more blood will go through the postcapillary venules. Blood flow to the area will increase. That's the key consequence of vasodilation. So this is going to lead to increased blood flow. So blood flow to the infected area or the inflamed area is going to increase. Now, why is that ideal? Well, remember, all of the troops are in the blood. If we have a faster rate of blood flow through the tissue, then the total number of troops that we can recruit from that blood that's flowing through the tissue into the extracellular fluid is now going to be greater. So increased blood flow means that we can recruit more proteins from the blood and more white blood cells from the blood into the affected area. So that's why we want to increase blood flow to the affected area. So that's one of the effects of the inflammatory response, but that's not the only part of the inflammatory response. So let's now go to another part of the inflammatory response. So now we're going to talk about the effect of the capillaries and postcapillary venules at the same time. And the reason is that as far as the inflammatory response is concerned, the capillaries and post-capillary venules have the exact same response. So we need to deal with the terminal arterioles separately, but for the capillaries and post-capillary venules, we can treat them together. Okay, and two things are going to happen to the endothelial cells of the capillaries and post-capillary venules. And remember, the post-capillary venules are just like giant capillaries anyway, so it makes sense that potentially their response is going to be similar to the capillaries, and indeed it is. So in the capillaries and the post-capillary venules, you're going to get two things happening to the endothelial cells. Number one, you're going to get something called endothelial cell, and I'll just abbreviate endothelial cell to EC. Endothelial cell contraction, and this is what's going to allow us to recruit proteins from the blood into the extracellular fluid of the underlying connective tissue of the epithelium and the extracellular fluid of the epithelium itself. So let me draw a picture to show you what I mean by endothelial cell contraction. So this will require some basement membranes. So again, we get our evil red back again, we get our thick pen, and let's just draw some basement membranes here, and I can get rid of that horrible mark that's there at the same time, and I haven't managed to, there we go. We'll make a very thick basement membrane here. So here's one basement membrane. I'm just going to draw another basement membrane here. Right, so let's say both of these are basement membranes of one of these microvasculature blood vessels, so a capillary or a post-capillary venule. And now let's just draw some endothelial cells sitting on them. So here, whoops, I need to go back to the thin pen. Here is an endothelial cell sitting on this very thick basement membrane, and here is another endothelial cell sitting on this very thick basement membrane. So this is before endothelial cell contraction has occurred. This is the before picture. So we've got a basement membrane, and we've got these endothelial cells sat on the basement membrane, and you'll notice that at the junction between the endothelial cells, they're nicely opposed to one another. They've sealed this gap in between them. So there are lots of molecules between the membrane of this endothelial cell and this endothelial cell that are holding them very close together, so that this is effectively sealed to stop things coming from the lumen of the blood vessel out into the extracellular fluid. And remember, that's what this picture is effectively showing. This is the lumen, this is the extracellular fluid, and now we've sealed the gaps between those endothelial cells there. Now, we're going to now go over to the after picture. So the picture below is going to be the after picture. So interleukin-1, TNF-alpha, and histamine are going to come along to the endothelial cells of the capillaries and the post-capillary venules. They're going to work on receptors on the surface of the endothelial cells, and they're going to trigger changes in these endothelial cells, and this change is going to be endothelial cell contraction. Now use your imagination. What do you think endothelial cell contraction is going to involve? Well, indeed, it involves the endothelial cells getting smaller. They withdraw their peripheries in, they contract, just like a muscle cell would contract, like so. So these endothelial cells go from looking like this to looking like this. They withdraw their peripheries in, they contract. And what does that mean? It means that they now open up gaps in between them where potentially fluid from the blood and also proteins from the blood can move out of the blood and into the extracellular fluid. And this is exactly what can now happen. So, fluid from the bloodstream along with proteins from the bloodstream can leave the bloodstream through these gaps between the contracted endothelial cells and go into the extracellular fluid of this area where the inflammatory response is occurring. And that fluid that you now bring into the extracellular fluid of the affected area is called an exudate. So when you have a fluid coming out of the bloodstream that contains all of these inflammatory proteins, these antimicrobial proteins, that is referred to as an exudate. Whereas when you just have fluid that is normally coming out of a blood vessel, because of course you have a little bit of exchange between the fluid of the bloodstream and the fluid of the extracellular fluid normally in capillaries that aren't undergoing the inflammatory response, that's called a transudate that you're producing. So exudate and transudate are different. When you produce fluid with loads of antimicrobial proteins from the bloodstream as part of the inflammatory response, this is called an exudate. So, from both the capillaries and the post-capillary venules, you will get oozing out of the bloodstream this exudate, which is just fluid with loads and loads of antimicrobial proteins in it. And this is why inflamed areas of the body where the inflammatory response is occurring, tend to swell. So actually, I'll just go over the pillars of inflammation, the key characteristics of inflammation. So, when an area becomes inflamed, there are a bunch of key characteristics that are often pretentiously referred to as the pillars of inflammation. They're the key characteristics of an inflamed tissue. Now, one of them is that the area becomes red. And they're often given in Latin just to be pretentious. So the reason the area becomes red is that you've got increased blood flow. So increased blood flow makes the area red, and we refer to this as rubor. So this is one of the pillars of inflammation, rubor. The inflamed area becomes red. In addition, the inflamed area becomes hot. That is, in Latin, referred to as calor. So calor refers to the area becoming hot. Again, that is because of the increased blood flow. If you've got more blood is hot, flowing through an inflamed area, that will make the area hot. So rubor and calor are two of the four major pillars of inflammation. Then, another one of the pillars of inflammation is that the area becomes swollen, and this is due to the exudate that is forming, this more fluid coming into this area that contains all of these antimicrobial proteins that are hopefully going to start attacking the pathogen that is present in the area. But as I say, this isn't particularly powerful in the fight against influenza. It's more powerful against certain bacterial cells if they were invading the area. So, in Latin, the word for swelling is tumor. So, rubor, calor, tumor, they're all pillars of inflammation. The final pillar of inflammation is pain, which is dolor in Latin. And we'll talk about where pain comes from later on. It's going to come from the prostaglandin E2, but we haven't got there yet. We'll come on to that. So those are the four old pillars of inflammation. Some people add a fifth pillar of inflammation. It wasn't in the original description of the pillars of inflammation, but often people put a fifth one, which is loss of function of the affected area, which I think in Latin is functio lesi, and I won't attempt to spell that in Latin, so I'll put that one in English. So the final pillar of inflammation is function loss. So you lose the ability to actually use the area, the tissue that is undergoing the inflammatory response. So as I say, these are referred to as the pillars of inflammation here. They are just the characteristics of inflammation, the characteristics of an inflamed tissue. So these are the 5 or 4 pillars of inflammation if you want to miss out this newer one, function loss. Okay, right. So, continuing on back with the story, that was just a little aside, another little aside of this video. So, interleukin-1, TNF-alpha, and histamine, they've acted on the endothelial cells of the capillaries and the postcapillary venules, and they have caused those endothelial cells to contract like so, and this has led to the development of an exudate at the area. And remember, the function of this exudate is it's bringing in loads of troops that are going to help, hopefully, annihilate the pathogen, proteins that attack the pathogen. And I might just write that down here. So the function of an exudate, and let's just change colour to be colourful. The function of an exudate is that we are bringing in antimicrobial proteins that are hopefully going to help us annihilate the pathogen. Loads of antimicrobial proteins that were circulating in the blood, doing nothing previously, but now have been brought out into this area to actually perform the role that they were created for. So, that is happening in both the capillaries and the postcapillary venules, remember. So, the final major thing that's going to happen as part of the inflammatory response is the recruitment of white blood cells. So remember I said there were 2 major types of troop in the blood. There were the antimicrobial proteins and then there were the white blood cells. We've talked about how we're going to recruit antimicrobial proteins. We now need to talk about how we're going to recruit white blood cells. So, and the proper term for white blood cells is leukocytes. Leuk, remember, means white. So these are white cells, white blood cells. So we're now going to talk about leukocyte recruitment, which is another part of the inflammatory response. So again, this is carried out by the endothelial cells of the capillaries and the postcapillary venules. So let's just draw a little bit of a picture of this. So again, we're going to need a drawing of the endothelium here. So here is our basement membrane again, and I'll just go back to the thin pen. And here are our endothelial cells that have undergone the contraction process here, so they've opened up these gaps between them, and the white blood cells are going to come through the gaps between the endothelial cells. However, unlike the fluid and the proteins that form the exudate, they cannot just come through the gaps unassisted. They have to really squash and squeeze to get through this gap, and in order for them to do that process, they need to receive a lot of help from the endothelial cells. These endothelial cells need to put loads of special proteins on their surface that can interact with a whole bunch of proteins on the surface of the white blood cell, so that the white blood cell will undergo this process where it squeezes through here. So just to draw this on, let's put our white blood cell, and because I've already used white for the endothelial cells, I can't put the white blood cell actually in white, but they're not actually that white. So here, this can represent our white blood cell, and it's a very basic shape that I'm drawing. This one would actually represent maybe a monocyte in the blood, which is the precursor to macrophages. So let's say this is a monocyte within the blood, which is one of the types of white blood cell that's going to be recruited early on in the inflammatory response. So this is terribly relevant that we're discussing this one. So here, this monocyte has started interacting with the surface of this endothelial cell here. What now needs to happen is it will adjust its shape and it will start squeezing through this gap like so. So here, I'm drawing it squeezing through the gap, and this requires this interaction with loads of proteins on the surface here, and I'm not going into the detail of what those proteins are. I'm leaving it vague because, you know, this isn't a video on the inflammatory response, this is a video on influenza, so we don't need to go into the details of how this works, we just need the basic overview. So, the monocyte is interacting with molecules on the surface of the endothelial cell, and then it moves over here into the gap. It'll start interacting with ones on this endothelial cell as well, and then it starts changing its shape, which will require cytoskeletal rearrangements, and it slithers like a slug through this gap here, and then it will return to its original shape on the other side, and it will differentiate into a macrophage. So it will become a great blob of a cell here. So here we go, this is our monocyte arriving in the underlying tissue, and it now differentiates into a macrophage. Some of the monocytes may differentiate into dendritic cells. Monocytes can become macrophages or dendritic cells, but most of them are going to become macrophages, which is why I'm drawing a macrophage here. Now, this process of a white blood cell slithering through the gap in between two endothelial cells has a special name. It is called diapedesis, spelt like so. So, in order for the endothelial cells to participate in leukocyte recruitment like this, they have to put loads of molecules on their surface. What makes them put those special molecules on their surface? They don't normally have them there. It is these molecules again, interleukin-1, tumor necrosis factor alpha, and histamine. So these molecules, interleukin-1, TNF-alpha, and histamine, they act on the endothelial cells of the capillaries and postcapillary venules, and they cause two things to happen: endothelial cell contraction, and then also the putting onto their surface of loads of special protein molecules that are going to function in leukocyte recruitment. And this means that they create an inflammatory exudate at the site, so they bring in loads of these antimicrobial proteins from the bloodstream into the affected site, and they also bring in loads of white blood cells into the affected site that are going to help fight the pathogen as well. Okay. So, there we are, we have discussed the inflammatory response. We still have more to discuss, of course. Where is all of this exudate and these white blood cells going to go? They're going to go into this underlying connective tissue here, and they're also going to go onto the actual epithelium. So in the epithelium, you're also going to get exudate, you'll get the fluid that has come from the blood with the antimicrobial proteins leaking out onto the epithelial surface here, and you'll also get the white blood cells appearing on the epithelial surface. So they don't just go into the underlying connective tissue, they also actually go onto the epithelial surface as well. So, what are we going to discuss next? We are going to discuss which types of white blood cell are actually going to be brought in. We've discussed one of the types, which is macrophages, but there are two other types that I want to discuss. And this is important because unlike the exudate, which doesn't really have that much effect on the influenza viruses, the white blood cells are going to help with the fight against the influenza virus. They're going to begin the fight against the influenza virus. Yes, in order to actually bust the influenza virus out, we're going to need more powerful white blood cells, the white blood cells of the adaptive immune response, whereas at the moment we're just discussing the white blood cells of the innate immune response, but these are going to have an effect, and I want to discuss that. Then we'll discuss interleukin 6 and prostaglandin E2 as well, and then we'll go on to discuss the local symptoms, the production of the local symptoms and the production of the systemic symptoms. Okay, so what types then of white blood cells are going to be brought in? So one of them I've put here. We're going to bring in macrophages, and these come from monocytes. Remember, there are not macrophages circulating in the bloodstream. We have these precursors to macrophages called monocytes circulating in the bloodstream. When they leave the bloodstream and come into some peripheral tissue, then they differentiate either into a macrophage or a dendritic cell. Usually they differentiate into a macrophage, although some will differentiate into dendritic cells. So we're going to bring in loads of macrophages into our epithelium that is infected. So we're going to get loads of macrophages appearing in that underlying connective tissue of the epithelium and actually appearing in the epithelial layer itself. What other types of white blood cell are we going to bring in? Well, hopefully, if you know any immunology at all, you should be able to tell me one at least here now. I need two that I'm thinking about here, but one of them you should be able to tell me, which is the neutrophil. So let me draw this here. So we'll have the neutrophil shown in yellow here, and it is, if you like, the cheapest type of white blood cell. You have loads of neutrophils. If you look at all of the white blood cells in the bloodstream, most of them are neutrophils. I think usual concentration of neutrophils within the bloodstream is between 2.5 and 7.5 billion cells, so times 10 to the 9, so billion cells per litre. So if you take a litre of blood in a normal person, then you will find, if you count every single neutrophil in that you will have between 2.5 and 7.5 billion of the things in that litre of blood. So there are a lot of neutrophils in the blood. We have a huge number of them, and in the inflammatory response, the acute inflammatory response, they are the major cell type that you throw into that area to try and destroy the pathogen. Okay, so let's draw a picture then of the neutrophil. So remember, neutrophils have this characteristic strange nucleus. They only have one nucleus, even though they have this strange picture here. But the nucleus has multiple lobules. It is not a normal round nucleus. Instead, it is split up into these multiple lobes here that are connected by little stalks in between them. So neutrophils have multi-lobed nuclei, and the characteristic number of lobes that the neutrophil nucleus has is 3. However, it can be more than 3. It can be 4, it can be 5, it can even be 6. And for this reason, neutrophils have another name. They're also called polymorphonuclear cells. And of course, the meaning of that is poly means many, morpho means shape, nuclear means referring to nucleus. So they are cells that have many shaped nuclei. They're the cells that have very variable shaped nuclei, so polymorphonuclear cells, and this is often just abbreviated down to PMNs. So that's another name for neutrophils. So these will be recruited from the bloodstream as well by these endothelial cells that have been acted upon by interleukin-1, TNF-alpha and histamine. And by the way, just to make this absolutely clear, these changes are the inflammatory response. The endothelial cells, they are the things that undergo the inflammatory response. Interleukin-1, TNF-alpha, histamine, they cause these changes in endothelial cells. That is the inflammatory response. And then the endothelial cells, by undergoing the inflammatory response, lead to the production of the exudate and the recruitment of the leukocytes, etc., and the vasodilatation of the terminal arterioles. But fundamentally, the inflammatory response is something that the endothelial cells undergo. It is a response in the endothelial cells to these cry for help that comes from the sentinel cells, interleukin-1, TNF-alpha, and histamine. Okay, so back to neutrophils. They come in again through being recruited by the endothelial cells that have undergone the inflammatory response. They diapodese their way through, and then they arrive in this connective tissue underlying the respiratory epithelium, and then they can move through onto the epithelial surface as well. So, before we talk about the third type of cell, let's just say what macrophages and neutrophils are actually going to do. Both of these types of cells, macrophages and neutrophils, they are both phagocytes. And that means that they undertake the process of phagocytosis. Now, the process of phagocytosis is where you engulf a pathogen or some other particle, some very big particle. You engulf it into an internal membrane-bound compartment. And indeed, phagocytosis is actually a form of endocytosis. Remember, endocytosis is where you take a piece of the plasma membrane and internalize it so that you have then an internal membrane-bound compartment. Phagocytosis is a form of that, and it's triggered by the cell wanting to internalize some massive great particle, potentially an entire pathogen. So you get this internal membrane-bound compartment containing the pathogen, and I might just try and draw this here. So let's say we have an influenza virus here next to the macrophage. What the macrophage can do is it can phagocytose that, and what happens is it brings this particle into an endocytic vesicle, effectively, although it's not called an endocytic vesicle anymore. I'll get some colour to put it in here. So there's the influenza virus particle, and here's the membrane around it. That membrane-bound compartment is now called a phagosome rather than an endocytic vesicle, and the reason it's called something different is that it's far, far bigger than any endocytic vesicle would ever be. You know, this is a massive great particle that it has potentially internalized there. Of course, in this case, it's internalizing a tiny little virus, but it could be internalizing a whole bacterial cell, which would be far bigger. Okay, so you get this this particle, this pathogen particle, internalized into a phagosome, and then what happens is it goes through something very similar to the endocytic pathway. It fuses lysosomes with the phagosome. So I might just draw this on here. So here are some lysosomes. These lysosomes will fuse with the phagosome, they'll release their lytic enzymes into the phagosome, and they will break down the particle. So this is the process of phagocytosis. You internalize some massive great particle, potentially, that you believe is a pathogen, and then you fuse lysosomes with that phagosome to destroy the thing inside. And this is what neutrophils and macrophages do. Now, neutrophils are quite bad at doing it. They're not as good as macrophages. Macrophages are a much stronger cell than neutrophils. Neutrophils generally phagocytose one great big particle, and then they give up, they die. All of their energy is put into killing this thing that they've internalized, and then after that they end up dying. And it's mainly because the horrible mechanism that they use to try and kill the thing inside the phagosome consists of more than just the lysosomes. I'm telling you a very simple story here, that they fuse lysosomes with the phagosome and that's how they kill the thing. They have a bunch of other horrible mechanisms that they unleash on this phagosome, and unfortunately those horrible mechanisms spill out onto the rest of the cell and therefore can end up destroying the cell in the process of trying to destroy the thing inside the phagosome. And neutrophils generally do end up dying after phagocytosing one thing, whereas macrophages are stronger. They can survive the phagocytosis process and then they can go on to phagocytose something again. Okay, so overall, what is the key message of that great big monologue there? Well, you bring in these two types of cell that are both phagocytes, neutrophils and macrophages. They're coming into the respiratory epithelium and they're now going to start phagocytosing the viral particles. If they find influenza virus particles, they will phagocytose them. In addition, they're going to phagocytose the dying cells. Remember, the host cells that have been infected by influenza viruses, after they've produced loads of new influenza viruses, they die. Those dying cells, they have to be cleared by something, and that's going to be the job of the phagocytes, the macrophages and the neutrophils. So let me just jot some of this down here. So, how are these two cell types going to help with this ordeal? They are going to phagocytose the viral particles directly. So they can phagocytose the virus, which is very useful because that's going to destroy virus particles, fantastic. But they can also phagocytose the dying epithelial cells. And I'll just abbreviate epithelial cells to ECs. And I know I use ECs for both endothelial cells and epithelial cells, but I'm sure you can cope with that. Okay, so those are two of the cell types that we are bringing into the respiratory epithelium, macrophages and neutrophils, and their role is phagocytosis. They will phagocytose influenza virus particles and the dying epithelial cells, and that's how they're going to help with things. Because, of course, the dying cell will still contain some virus particles inside of it, so it's helpful to phagocytose those cells and destroy them. Right. So there is another type of cell that's very important that is going to come in as part of the innate immune response. Obviously, there's going to be loads of white blood cells that are part of the adaptive immune response, but we'll come on to those later. We're just doing the innate response at the moment. So let's get some other colour here. So I have mentioned these cells earlier on, so we'll have a mustardy yellow colour back again for these types of cells. They are the natural killer cells. This is the other third type of cell that we're going to bring in here. So I'll draw one of these here. So it's a nice, simple, round cell with a big nucleus here that has a similar structure to the lymphocytes, so the B cells and the T cells, but of course they are of the adaptive immune system, whereas these cells are of the innate immune system. So this is a natural killer cell, also referred to simply as an NK cell. And as I say, these are going to be brought in as part of the innate immune response, and they're going to end up in the connective tissue underneath the respiratory epithelium and in the actual respiratory epithelium itself. So, what do natural killer cells do? Well, these are cells that can sense cells that are infected by viruses. They go around looking for virally infected cells, and if they find a virally infected cell, they will kill that virally infected cell. Now, how do they tell whether a cell is virally infected? Now, there is another great big part of the immune system that has the job of detecting whether cells are virally infected, and that is the cytotoxic T lymphocytes. But they are part of the adaptive immune response, and they use the major histocompatibility complex class 1 I mentioned earlier on. Now let me just find where I put this stuff. Here we go. So remember, I mentioned this molecule, major histocompatibility complex class 1, which is really important for telling the immune system, the adaptive immune systems, telling cytotoxic T lymphocytes, which we will come on to much, much later when we do the adaptive immune response, telling these types of cells whether the cell is infected with viruses or not. However, that's a very complicated mechanism. That's part of the adaptive immune response. These natural killer cells, they are going to look for virally infected cells by looking for lack of MHC class 1. Remember we said that influenza virus is clever. It creates this non-structural protein 1, and this non-structural protein 1, along with stopping the production of the type 1 interferons, lowers the production of the major histocompatibility complex class 1. And the idea is that if it lowers this, it protects itself from the adaptive immune response. It protects itself from cytotoxic T lymphocytes, because without this, the cell can't tell the cytotoxic T lymphocytes that it is infected by the virus. So the virus thinks, brilliant! I've come up with this fantastic mechanism to avoid the adaptive immune response, to avoid these incredibly scary cells called cytotoxic T lymphocytes. However, the human body has fought back. It's come up with a response to this. It has created these natural killer cells that are part now of the innate immune system. They aren't specific at all to what virus they're dealing with, and they go around looking for cells that have got lower MHC class 1. And if they find a cell that has far too little MHC class 1, they say, you are misbehaving. Why are you misbehaving? It's probably because you've got some clever virus, such as the influenza virus, that is making you make less MHC class 1 and put it on your surface. And it's not just the influenza viruses that have mechanisms to reduce the MHC class 1 molecules on the surface of the cell. Other viruses deploy this tactic as well. So, the natural killer cells are the human body's response to this mechanism of the virus. And as I say, they will go around and look for cells with low MHC class 1, and if they find them, they will kill them. So let's go into the details of how they do this. So here we go. So, natural killer cells on their surface have a protein, a receptor, called the KIR receptor. So this is spelt K-I-R, the KIR receptor. Now what does this stand for? This stands for killer cell, that's the K, and of course that means referring to the natural killer cells, the NK cells. So killer cell, and then the I is for immunoglobulin, which I'll just abbreviate down to IG, like receptor. So this is the killer cell immunoglobulin-like receptor. And the bit in the middle, immunoglobulin-like, it just means that this receptor bears similarities to some of the immunoglobulin some of the immunoglobulin domains in immunoglobulin molecules. So if you look at the heavy chains and light chains of immunoglobulin molecules, they have special domains in them, special little patches of the protein that are called immunoglobulin domains. So this receptor will have some domains like that. So this is to do with the structure of this protein. That's the reason that that's there. The other bits make perfect sense. Killer cell, it's obvious what that means. Receptor, we know what that means. You might wonder what this means. It just is referring to the structure of this thing, so don't worry about that. The key message here is that we have this receptor on the surface of natural killer cells called the killer cell immunoglobulin-like receptor. And this is a receptor that can bind to MHC class 1. So the ligand for this is MHC class 1. So what happens is natural killer cells, they go around and they look at cells. They get nice and close to cells. So I might even draw this here. So let's say we have some peripheral cell here. So this is the cell that this natural killer cell is now coming to examine. And what it will do is it will go up to this cell and it will want a lot of its killer cell immunoglobulin-like receptors, its KIR receptors, to bind to MHC class 1 molecules on the surface of the normal cell. So let's say that these purple blobs that I'm putting on the surface of the cell here, these are MHC class 1 molecules, so they're all MHC class 1. So let's say this cell isn't virally infected and has a lot of MHC class 1 on its surface. What will happen is the KIR receptors of the natural killer cell, they will bind to the MHC class 1 and they will now deliver an inhibitory signal to the natural killer cell. These will inhibit the natural killer cell and they will say, do not kill this cell. This cell is behaving perfectly. It has loads of MHC class 1 molecules on its surface, so it's telling the adaptive immune response exactly what we want to know, and it's telling the adaptive immune response at the moment that it's not virally infected. It's got these MHC class 1s. If it's not virally infected, then those MHC class 1s will be telling the adaptive immune response that it's not virally infected. But our job is just to check that it has enough MHC class 1s on its surface. Okay, so this will inhibit the natural killer cell, and the natural killer cell will not kill this cell. If, however, it comes up to a cell that is virally infected by one of these clever viruses, and therefore has hardly any MHC class 1s on its surface, then it will not get its KIR receptors binding to loads of MHC class 1s, and that will mean that it doesn't get this inhibitory signal, and then it thinks, uh-oh, this cell does not have enough MHC class 1, it knows that now, and it's going to unleash the killing mechanisms on this cell. So what are natural killer cell killing mechanisms? Well, they use the perforin granzyme killing mechanism, which is actually a killing mechanism also deployed by cytotoxic T lymphocytes. So what they're going to do is they're going to release granules inside their cytoplasm. So I'll just draw one of these. So inside the cytoplasm of the natural killer cell, you have granules, vesicles containing horrible, horrible proteins. So these are the natural killer cell granules. So what they're going to do is they're going to release these onto the surface of the cell that they do not like because it doesn't have enough MHC class 1. So bring them over here, release them onto the surface of this cell, and what is actually in these granules? There are two molecules that are really important, perforin and granzyme. So let's just get some interesting colours to do these in. So we'll have perforin here in green, so this protein can represent perforin, or this blob can represent perforin. So that's perforin, and then we'll have granzyme in blue. and granzyme is an enzyme. So in blue here, this is granzyme. So in the granules of the natural killer cells, you have perforin proteins and granzyme proteins, and these two are going to work together to destroy this cell that the natural killer cell is next to and doesn't like. So, what do the perforins do? They go into the cell membrane of the cell and they oligomerize, they join together into an oligomer, which is a complex built out of some proteins. So it's similar to a polymer, but remember, polymer means you've got loads of separate subunits that you've joined together, whereas oligomer means that you've got a few subunits that you've joined together. So perforins are going to oligomerize and they form channels in the cell membrane. So I'll draw one of these here. So it's formed a channel in the cell membrane. Actually, I'll draw it better. I'll draw it in the more conventional way to show a channel like so. Okay, so lots of perforins join together to form this channel. So you'll form these channels in the cell membrane. And of course, you won't just make one of these. You'll make loads of perforin channels in the membrane of this cell. Now, what do these channels allow to move into the cell? They allow the granzymes to move through. So this is why they work together. The perforins create a channel through which the granzymes can then go into the cell. And once the granzymes are in the cell, they set off all sorts of catalytic reactions. So they set off a cascade of reactions that leads to the apoptosis mechanism being initiated inside this cell. So this cell now undergoes apoptosis, which is programmed cell death. So that is cell suicide. Contrast that to the type of death that the cells that are infected with influenza viruses undergo. They undergo accidental cell death. They undergo necrosis. In fact, I might just write that down. Where are we? Up here, where this cell died, it underwent the process of necrosis. It did not plan to die. It did not initiate the apoptotic mechanism. It did not kill itself in a beautiful, coordinated way. It was killed by the strain of making all these influenza virus particles. Whereas the granzyme is going to initiate a cascade inside the cell that will initiate the apoptotic mechanisms, and therefore this cell will self-destruct. Okay, and that's how natural killer cells are involved in the innate immune response. This is a totally non-specific response. It does not need to know anything about the pathogen it's dealing with. All it does is go around looking for cells that are abnormal, that have low levels of MHC class 1, because if you have low levels of MHC class 1, you are not behaving, you are potentially a very bad cell, potentially because you're virally infected, or for more sinister reasons as well, such as cancerous cells can start lowering the level of MHC class 1 on their surface. So NK cells aren't just about killing virally infected cells, we don't think. They may well have a role in killing cancer cells as well. They go around looking for cells that are misbehaving with too low MHC class 1 molecules on their surface. So, overall, these are the three types of white blood cell that we're going to bring in then. Neutrophils, macrophages, and natural killer cells. Neutrophils and macrophages are both phagocytes. They're going to phagocytose viral particles, and they're also going to phagocytose the dying columnar epithelial cells. Natural killer cells, meanwhile, they're going to go around and start killing virally infected cells. They're going to look for cells that have too low levels of MHC class 1 because those are bad cells that are virally infected, and they will unleash these horrors within their granules, the perforin and granzyme proteins. Perforin oligomerizes in the cell membrane to create a channel. Granzymes then go in through the channel into the cytoplasm of the cell, set off cascades inside the cell that leads to the initiation of the apoptotic mechanisms, and the cell commits suicide, it dies. Okay, so that is the leukocytes and how they are going to function in the inflammatory response. So that is how we are going to try and attack the influenza viral infection using the innate immune response, the inflammatory response. Let's now just finish the inflammatory response by talking about what these other molecules do. I'm not going to talk about interleukin 6 yet. I'm going to leave off on that one for a while. We'll come on to that when we discuss systemic symptoms, where the systemic symptoms come from, because that's where interleukin 6 is really important. Let's just talk about prostaglandin E2, what this does. So prostaglandin E2, you could call this the pain molecule. This is the molecule that we believe underlies inflammatory pain. So when you have a portion of tissue that is inflamed, that tissue is painful. Dolor is one of the pillars of inflammation. That type of pain is called inflammatory pain. You can contrast that to pain that is caused by pressure, when something is putting a lot of pressure on a certain portion of tissue, or pain that is caused by acid, or pain that is caused by the pain nerve cells misbehaving, neuropathic pain. They might start firing for no reason because they're broken. Those are different forms of pain. Prostaglandin E2 is believed to underlie inflammatory pain. So I'll just write this down. So this is the molecule that mediates inflammatory pain. So how does this work? Well, in most tissues of the body, not all tissues of the body, but most tissues of the body, you have pain receptors. And these are called nociceptors. So let me draw one of these. So the nociceptor is the name for the structure at the end of the neuron that actually conducts the information back into the spinal cord. So just drawing this here. Here is the neuron, and let's say here is its cell body that will be in the dorsal root ganglia of the spinal cord. And here is its central process that will be going into the spinal cord. So I'll just actually draw the spinal cord here. So this column here, this is representing the spinal cord. Here is one of these pain neurons, and to give it its full name, and I don't know if I've got room to write down its full name, luckily this extends along here. So to give this neuron its full name, it is a primary nociceptive, which means pertaining to pain, or pain perception, primary nociceptive afferent, and that means that it's conducting information into the central nervous system rather than out of the central nervous system. Primary just means it's the first neuron along, and it will synapse onto a secondary neuron that will then carry the information up to the brain. But this is far too much neuroscience. We don't need to go into all this detail. So this is a primary pain neuron. Now, on its peripheral process, so it's got two processes, as you can see. It's what we call a pseudo unipolar neuron. So yeah, we are going into neurobiology here. So this is described as pseudo unipolar, and the reason it's called that is off its cell body. You can just see the way I've drawn this. Here's its cell body where the nucleus will be. Off its cell body, you only have a single process coming off. Now, when a neuron only has a single process, we call it unipolar. However, this single process is splitting into two processes that are going in opposite directions. So it is effectively actually a bipolar neuron with two processes, a central process and a peripheral process. And this one here that goes into the central nervous system, the spinal cord, that's the central process, and the one that goes off to receive the information from the far off peripheral tissue, this is called the peripheral process. So effectively it is a bipolar neuron, although we can't strictly call it a bipolar neuron because for it to be a bipolar neuron, the two processes have to come directly off the cell body, which they don't. They come off this process that has come directly off the cell body. And that's why we call it pseudo-unipolar. It's a fake unipolar neuron. It's like a bipolar neuron, but it's not, so we call it pseudo-unipolar. So, this is our primary neuron, which is a pseudo-unipolar neuron. It has the peripheral process, which collects the information about pain signals in the peripheral tissue that it innervates and takes the signal up. Then it will go down the central process, will release neurotransmitter onto a secondary neuron that will then carry the information up to the brain. Okay, brilliant. What we now want to discuss is what is it that's on the end of this peripheral process. So, this peripheral process is in the peripheral tissue. It might be innervating, let's say, the back of our throat potentially, so the respiratory epithelium of the top part of the nasopharynx, let's say. So it's in the underlying connective tissue, so go back to this picture. It might be in this process. What is actually on the end of this peripheral process then? It's what we call a nociceptor, and it looks like a little tree. So this structure here, this is the actual nociceptor that will be in the peripheral tissue and which is collecting information about painful stimuli. So this portion, this tree portion, is what we actually refer to as the nociceptor. It is the pain receptor on the end of the pain neuron. So on the end of the peripheral process of this primary nociceptive afferent, you then have the nociceptor, the pain sensing structure. So what happens is painful stimuli such as pressure or acid or heat too high temperature or too low temperature or for instance this horrible molecule prostaglandin E2, they activate receptors in this nociceptor, this tree-like structure which is all membrane bound and is part of the cell, and those will start to produce electrical signals that will all sum together to produce an action potential here. So they'll produce depolarization, activation of the receptors on the nociceptor for the noxious stimuli, the painful stimuli, cause depolarization at the site of the receptor. That depolarization can sum together at this point here where the nociceptor junctions with the peripheral process here, and then if that depolarization is big enough, you'll fire action potentials which will go along the peripheral process, along the central process, and cause the release of neurotransmitter, activating action potentials in the secondary neuron. That will go up to the brain, and the brain will say, ow! Okay, that's the signal that causes the brain to feel pain. Okay, right. So, all of this discussion, why have I gone into it? Because prostaglandin E2 works on receptors that are in the membrane of these nociceptors that innervate tissues and causes the pain neuron to fire action potentials and therefore leads to the sensation of pain in the local area where it is present. Prostaglandin E2 is produced in inflammation and it causes inflammatory pain. So even if you don't understand any of the neuroscience, know that where you have this molecule produced, that produces pain in that area. It is a nasty, nasty molecule, prostaglandin E2, and the non-steroidal anti-inflammatory drugs, the NSAIDs, as well as paracetamol, the way that they produce analgesia from inflammatory pain is they stop the production of this molecule, prostaglandin E2. And that's why they're not so good at producing analgesia against other forms of pain that aren't inflammatory pain, because the way they work is by stopping the production of this molecule, and this molecule mediates inflammatory pain, not temperature pain, not acid pain, and not pressure pain. To relieve those form of pain, you need different types of painkiller that work, for instance, by blocking the signaling between the primary pain neuron and the secondary pain neuron, which is how the opiates work, like codeine, morphine, etc., things like that. Okay, but we'll come on to that later on when I want to discuss the treatments that you can take for flu. Right, so I think we will have a break at this point. We have now completed my discussion of the inflammatory response. I still want to go over symptoms with you though. I want to talk about where the local symptoms come from and where the systemic symptoms come from, and that's quite a fun bit, so we can look forward to that in the next video, but let's have a break now.